Good afternoon, everyone, and welcome to today's webcast titled Modeling Nutrients in Rivers and Streams, Lakes and Reservoirs, and Estuaries. This webcast is sponsored by EPA's Water Quality Modeling Workgroup. My name is Jason Gilday. I'm with US EPA, and I will be moderating today's webcast. And thank you for joining us today. First, I'm going to briefly summarize some of the features of today's webcast. One, we hope that you have a lot of questions, and please type your questions in the questions box on your control panel and click send. If your control panel is not showing, simply click on the small orange box with the white arrow to expand it. If you have any problems viewing the broadcast, just let us know in the questions box, and we will post an answer in the same box. A phone number is also available on your control panel if you are having trouble hearing audio through your computer speakers. This webcast will be archived for later viewing on EPA's Water Quality Modeling Workgroup page, and we will provide that link for you on a slide at the end of the webcast. Also, at the end of today's webcast, you will be able to obtain a certificate of attendance for participating, and we'll provide you directions for downloading it at that time. The materials in this webcast have been reviewed by US EPA staff for technical accuracy. However, the views of the speakers are their own and do not necessarily reflect those of US EPA. Mention of commercial enterprises, products, or publications does not mean that US EPA endorses them. So now that we've completed kind of all the housekeeping items, I'm going to kick off today's webcast by quickly going over the purpose and the history. This webinar series is brought to you by EPA's Water Quality Modeling Workgroup, and we are a group of EPA staff members at headquarters and throughout the regions who use models to solve water quality problems. Most of us are in the TMDL or standards program. Our group was formed kind of as a grassroots effort to facilitate collaboration among EPA staff who use models. The purpose of the webinar series is to help water quality professionals better understand water quality modeling and how models can be used to solve problems facing water quality regulators. The target audience for the series is a water quality professional who may use, want to use, or review models as part of their job, but who do not necessarily have a modeling background. As such, we're assuming that the audience has a basic understanding of water quality science and concepts, but has limited direct modeling experience. To reiterate from last time, there's a wide range of topics to discuss with water quality modeling, and this webinar series is presented as more of a broad overview or intro to water quality modeling. The webinars build off of each other, so I encourage people to watch the first 5 webinars if you can. All of the webinars will be recorded, archived, and posted to EPA's YouTube website. The first 5 webinars, actually I should say the first 4 webinars, are currently posted, and a link is available on our Modeling Workgroup website. The 5th webinar will be posted shortly. Today's webinar archive will be posted soon. It usually takes about a month to turn around the transcript and the YouTube processing. So today's webinar is the 6th in our webinar series and builds upon the information presented in the first 5. The webinar series has been constructed to begin with modeling basics, in other words, webinars 1 through 3, and then move into more specialized modeling topics. Your feedback on the series and content has helped shape each installment of the series. The last webinar covered aspects of nutrient water quality modeling and reviewed the nutrient cycle, impacts and implications of excessive nutrients in the aquatic environment, nutrients and mechanistic models, nutrient targets and endpoints, and how statistical relationships can link complex endpoints to model state variables. Today's webinar continues the topic of nutrient modeling and focuses more closely on nutrient modeling in relation to various water body categories, including rivers and streams, lakes and reservoirs, and finally estuaries. The 7th webinar in our series is tentatively scheduled to cover sediment water quality modeling, and we will send out an email when we have more information available on that. On a final note, before we kick off the presentation, we will try and answer as many questions as possible throughout the webcast. We'll be pausing periodically through the webcast today to do a variety of question and answer sessions. Please remember to type your questions in as the webinar progresses, and we will try to answer as many as possible. So to do a quick recap of the webinar so far, the first three presented a basic modeling overview and covered conceptual topics such as what is a model, why use one, types of models, how to select a model, steps in model construction, and interpreting model information. The fourth webinar started to get more detailed with a case study of temperature modeling with Qual2K. And of course, the last webinar introduced the nutrient water quality modeling that we're going to expand on today. So based on your feedback, we are continuing the focus on nutrient water quality modeling. Our presenter will be discussing various modeling tools and approaches for nutrients in different aquatic environments. He'll be talking about endpoints for nutrient modeling, and also he'll highlight some specific case studies today. Our speaker today is joining us again. He was a speaker last time, and I'm pleased to, I guess, reintroduce Tim again. Tim Wolf from EPA Region 4 is joining us as the speaker today. Tim is a national TMDL expert with US EPA from Region 4. He has over 25 years of experience in development and application of water quality models. Tim established the Watershed and Water Quality Modeling Technical Support Center located at US EPA's Office of Research and Development, ORD, in Athens, Georgia. And Tim is one of the primary developers of the Water Quality Analysis Simulation Program, WASP. Tim has supported US EPA with developing a number of technical approaches regarding nutrient numeric criteria in the state of Florida, and has significant experience in developing and reviewing TMDLs for bacteria, nutrients, metals, dissolved oxygen, and mercury. And with that, I'm going to pass over to control to Tim and let him get started.
Thanks, Jason. My hope today in this webinar is just to expose you to various modeling tools and approaches for nutrients for each of the typical water bodies that we're asked to model. We'll also look at the different water quality endpoints, whether they're standards or targets for TMDL development. We'll look at some of them. And at the end of each of the water body types, I'll talk a little bit about and present a little case study to show how the models can be used to provide us insight into how to control nutrients in the environment. So the major water body types that we're going to be looking at are going to be rivers, then lakes and reservoirs, and then estuaries and coastal areas. So we're going to first start off with modeling nutrients in rivers and streams. Just a little bit about the characteristics of rivers and streams. When it comes to the hierarchy of modeling aquatic systems, specifically for water quality modeling, rivers and streams are typically the, I'm not going to say the easiest to do, but they are by far a lot easier to parameterize a water quality model for. But rivers and streams are basically characterized by one-dimensional flow, and that varies based upon the size of the watershed. So water quality within the river and stream is controlled by the watershed that drains and comes into it. And when we talk about rivers and streams, we're talking about we can be doing small intermittent streams or first-order streams, second-order streams, all the way to large riverine systems like the Mississippi. Because rivers flow in one direction, it distinguishes us from all of the other water bodies we're talking about today. And this is one of the reasons why it makes it one of the easier types to model, because everything goes in one direction. It comes into your model domain and it leaves your model domain. So for us in rivers and streams, flow is like the major controlling or limiting factor in the rivers. So the more flow, the more things that can come into it. The less flow, the less that can come into it. So we expect flow to have a major impact on water quality, whether it's solution time, travel time in the system. We also assume that cross-sectional mixing is very, very rapid compared to the water bodies. Thus, we assume that the concentration within the river is going to be pretty much uniform across the river. So the factors that we're going to look at for this modeling approach is, well, we're going to look at, when we're looking at the model, we're going to look at flow complexity. So how complex is the system? How flashy is it? What kind of spatial resolution are we going to need to use in our model to adequately answer questions on the impacts of nutrients? We also have to consider whatever the endpoint is, or the water quality standard, because some of them are very, very simple and where they just basically say never to, and some of the other ones could have a frequency or a magnitude or a duration. So that would also drive what kind of modeling approach we should use. And then also the water quality model complexity depends on what kind of state variables in the model are we going to have to use? Are we going to have to use the full nutrient cycle, include things like multiple phytoplankton, benthic algae? We need to consider pH, alkalinity. All of that kind of controls what kind of modeling approach that we'll take. So as far as spatial resolution goes, can we just do a section of the river where we can actually parameterize what's coming into this section of the river from above, outside our model domain, to down below where we have to make our decision? Or should we just use do the whole watershed, including tributaries coming into the system? So it may be very difficult to determine in a section what will be coming in to our modeling domain. So we would have to expand and include the whole watershed. As far as temporal resolution, can we just do a critical condition? Can we just look at a growing season or a critical seasonal component? Or do we just need to do continuous simulation over annual or more than 1 year of simulation? So to use a steady state model for nutrients, it can be done. There's been several very good studies that a steady state model was used to look at impacts of nutrients on a water body. But to use a steady state model, you need to be able to define a critical condition. You know, whether that's a specific flow regime, temperature regime, light regime, and nutrient regime. One of the issues with using steady state for nutrients is that nutrients don't always express themselves at a single condition. It could be a low flow year followed by a high flow year, or vice versa, or it could be a unique set of flow temperature and light conditions that exist at different periods of time during the year. So for nutrient modeling, steady state's probably not the best approach. It certainly could be an initial approach for determining what are the important components for modeling nutrients in your particular water body. So when to use a dynamic model? Well, we're going to use a dynamic model when the flows are highly variable. And that could be anything from below peaking hydropower facilities or the impacts of transient inflows, such as stormwater events, variable point source discharges. Another reason to use a dynamic model is when the endpoints consider variability over time, like predicting or meeting a seasonal average, predicting impacts of storm events, or a subsequent period of time where there's no runoff from the system. When critical conditions are not known for your water body, where you have to actually use the model to go through series of it could be many, many years to determine where your critical conditions are and what are controlling them. And then ultimately, if what you're looking at to protect against is a frequency, magnitude, or duration of an exceedance of a water quality standard, you would have to use a dynamic model. So as far as water quality model complexities, the processes that you need to consider that are impacted by nutrients, Well, you have simple dissolved oxygen, and simple dissolved oxygen back in the day really only considered the impacts of BOD on DO concentrations. Now, we would kind of see what we would call a simple dissolved oxygen model now would include BOD, nitrification, and sediment oxygen demand. And that could be all you would need to do for a nutrient TMDL to meet a dissolved oxygen issue. Or you can look at doing intermediate eutrophication, where you're probably going to be doing a good bit of the nutrient cycling, but you may not be doing multiple phytoplankton impacts of benthic algae on water quality. And then when I say eutrophication, it can include any of the ones below it, where you're looking to control nutrients to meet a specific target or water quality standard for algal population. It may be critical to consider multiple algal components because you have different algal species in your water body that exhibit or could be controlling at different times of the year. Or you can have benthic algae, periphyton, or macroalgae that are influencing water quality quite dramatically. And then lastly, all the productivity could have an effect on pH and alkalinity in your system, and ultimately the water quality standard that's being violated is pH, while there may not be standards for algal population. So those are the things that would drive the model complexity. So for endpoints, well, the first one is dissolved oxygen. All the water bodies in the United States are going to have some kind of dissolved oxygen standard associated with them. So one of the endpoints could be a minimum, so not to ever go below a certain value. It could be you must have a daily average DO concentration, or you can set a frequency, which a lot of the site-specific DO standards are using now, where you set a frequency where the dissolved oxygen has to be above this value for aquatic species survival. So a minimum not to exceed, or not to go less than, I'm going to use a value of 4, a daily average of 5, and a frequency where it can't be below X value for more than 10% of the year. The next potential endpoint for rivers and streams is pH, which is typically influenced based upon productivity due to photosynthesis and respiration, which during large, highly productive periods of time, the pH can swing quite dramatically. So you may have to control nutrients to control productivity to meet your pH standard. One of the typical endpoints that's not really considered that often, there was a great benthic algae modeling study done by, I believe it was the state of Montana, and I believe even some of it was done on the Yellowstone River, where one of their endpoints was looking at nutrient impacts on benthic algae. Benthic algae, when in a nutrient-enriched environment, one can act like a biofilm and be a tremendous nutrient remover from the water column, but it also can grow in large abundances and have large impacts on dissolved oxygen, pH, and then ultimately have habitat alteration to remove suitable habitat from the bottom of the stream for macroinvertebrates and things like that. The next one is phytoplankton abundance, and a lot of the state's water quality standards for phytoplankton is that basically say not to be an imbalance of flora and fauna. So it's very, very difficult to define that in a sense, but we can come up with, through different studies, what would be a suitable chlorophyll concentration that would be representative of a balanced system. We also can look at phytoplankton as far as species shifts as the water body is exposed to different concentrations of nutrients. You can get species shifts from diatoms, blues to blue-greens. We also can look for nuisance and harmful phytoplankton blooms as endpoints. And then also, you could have endpoints that are protective of drinking water supply requirements to reduce the impact of disinfection byproducts. So you can actually come up with, going backwards from the drinking water supply to what would be an acceptable phytoplankton concentration that could be adequately treated without causing the disinfection byproducts. And then lastly, whether you're lucky or cursed, you could have a numeric nutrient criteria for nitrogen and phosphorus that also may have a chlorophyll concentration associated with it. So you could model your system to meet those numbers. Now, as far as dynamic analysis goes, you can do quasi-steady state approaches, and there are some steady state models that allow you to do quasi-steady state as well, where you can have steady flows and steady loads, but the model can make diel calculations, so you would get actual changes in dissolved oxygen over the 24-hour period of time, changes in pH and such. So you can start with a quasi steady state approach, but ultimately you're probably going to be going to a dynamic approach where you run a dynamic model with descriptive flows. And when I say descriptive flows, these flows could be coming from USGS gauging stations. They could be coming from a watershed model, and they could be estimated just in a spreadsheet to be put into it. So ultimately, you'll provide the model with predicting, giving a varying flow and concentrations coming into the model over time. For really detailed, where travel time is really critical, you may be coupling with a hydrodynamic and a water quality model to best represent the travel time in the system, because we'll see through the different case studies here, some of the critical components of getting a water quality model right is not only getting the loadings right, but getting the flows and the transport correct. Because once you have the transport correct, you have the travel time. Travel time is indicative of the retention time in the system, and as far as water quality responses to loads, it is a big function of how long it's in the system. So one of the things we have to think about is when and for how long do we apply our continuous simulations. And this is a tricky part, and this is actually was talked a little bit about, I believe, in webinar 3 or 4, but one of the things that we have to look at is we need to be making sure that we are covering, one, the period of time where we believe we have a problem, also a period of time where we believe we have adequate data to parameterize the model, and also constrain the model to show that the model is representing the system as best as we can. So, typical applications that require continuous simulation is when we have different conditions during wet and dry weather periods, where we have to consider the different loadings over time, followed by very dry periods. And again, we'll see some of that in our case studies. So the first case study is just going to be a simple one, just to show you what kind of information you can get from a time variable model. But the flows and loads were simulated in this model using LSPC, which is a watershed model, which is pretty much the same as HSPF, which is a widely used watershed model. So it's going to provide to, in this case, the loss model, inflows and loads, and we'll also integrate NPDES discharges into the model. And ultimately, our potential endpoints in this case study is we're going to look at dissolved oxygen, benthic algae density, and pH, because these are the dissolved oxygen and pH are typically the triggers in what we believe is the impairment in the system. Another interesting thing about benthic algae is in most cases where you have a benthic algae problem, you'll have pretty much low to no regular free-floating phytoplankton, because the benthic algae have this ability to luxury consume nutrients, and they can kind of outcompete the free-floating phytoplankton. So we just have this is our river system. The red area is showing where we have a water quality standards violation below the state's dissolved oxygen standard. And when we investigate this, we're going to look at a couple different things. The first thing is the model is going to be able to tell us, the first thing is this graph is actually showing you a couple days simulation of the DO. The red line is actually the dissolved oxygen standard. So ultimately, if we were to do a TMDL here, this was a never to exceed. We would have to do it to the point that all of the lines on the graph would be above the red line. But the top line is actually the maximum DO during the day. The middle blue line is the average DO, and then the black line is the minimum DO that was predicted during that day for the model. So we can, from the model, when it's well calibrated, we can see the diel swing of dissolved oxygen in the water body, and we can determine nutrient load reductions to meet the water quality standard. The next thing that we want to look at is, well, this is the benthic algae density. Now, this is one of the problems with benthic algae is a lot of states have just started to collect this, and it's very, very difficult to measure because ultimately we're going to be comparing this to grams ash-free dry weight per meter squared is what the model's outputting, and which is typically measured in the laboratory. So, but we can see what the densities are. States can develop targets for what is a protective density, and then you can reduce nutrients until you met that periphyton density. And then the last thing we wanted to look at is this is the pH prediction for the water body, and you can see that there is a large pH swing as a function of productivity in the system. So we would also develop nutrient reductions to control periphyton to meet the DO concentration and the pH concentration. With that, I'm going to stop and see if there's any questions related to streams.
Thanks, Tim. We've only got a couple questions so far, so maybe I'll just pause for a minute and remind people to get your questions typed in, and the questions kind of pop out. I'll kind of keep tally of these as we go on, so if we don't get it answered now, we'll try and get back to it later. And I see a few more coming in, so I'm just going to kind of launch into the first one, if you're ready, Tim. So the first question is, do dissolved oxygen models incorporate the indirect BOD, UOD from algae, parentheses, either systonic or otherwise? And as the asker is saying, it would seem that in some instances this could be the dominant source of BOD.
I think pretty much all models, all water quality models that consider algae do have the feedback as once the algae die, they actually go through the detrital phase where they have to go through this dissolution process, and then they are ultimately converted to dissolved organic carbon, which does impact dissolved oxygen. There are several water quality models out there that actually utilize more than one BOD component, so you can actually simulate how much BOD is coming from algae dying. So yeah, it could be, and it could be a major source, and it's not so much in rivers, but it can be a major source in lakes and reservoirs.
Thanks. Next question, what was the time period for the graphs that you showed?
Well, that simulation was actually, it's a 6-year simulation. I was just showing one period of time in, I believe it was July of 2008.
Okay. Are you considering benthic algae to be the same as periphyton?
Yes, yes, they are called. They are the same. They are attached algae.
We have one asker that's asked a number of problems, and I think he's getting at, are you going to touch on these? He has a list of them, including lakes and lines with reservoirs, floodplain wetlands, aquatic macrophytes. Maybe can you talk about how can you address kind of these complex issues, or are you going to touch on them later in your presentation?
I'm not going to touch on them too much, but I guess not going to talk too much about floodplain. Floodplains in river systems are kind of difficult because that takes it from a one-dimensional system to a two-dimensional system, and there's not a lot of models out there that can do that, because one of the problems is the floodplains do not have water in them most of the time. But there are ways, hydrodynamic models, that could be utilized to do that. And, one of them that's fairly good at it is HECRAS, which is a hydrodynamic hydraulic model. It does not have I know that they're adding water quality components to it, but it does not have a very robust water quality model associated with it yet. So I've been working with HEC to have them create a linkage file that WASP would be able to read, so we'll be able to consider that. The other tricky one in there is macrophytes, because macrophytes, some of the models can handle that they do macrophytes. And when we talk about macrophytes, there's kind of like there's two different types of versions of them. There are something that we call like macroalgae, which at times will start connected at the bottom, and then they would release and they kind of float around in the water. And then we have macrophytes that have roots and shoots, so they take their nutrients up from the sediment and from the water column. There are a couple of water quality models out there that can do macrophytes. Again, there's not a lot of data for them, though. Good question, though.
And I think in terms of lakes and lines with rivers, I know you're going to talk about lakes next in your presentation, right?
Yeah, lakes are next, yep.
Yep. Okay, a few more questions. What is your opinion on the application of Qual2K for benthic algae simulation under critical conditions? Maybe compare that to something like WASP.
Yeah, so I've had some interesting conversations with a couple of my colleagues in other regions about that. I believe that if we're talking about the application that was done in Montana, that was, I mean, they did everything you could get out of a steady state model. I thought it was a fantastic application. I had talked to the people that developed that application, and I kind of encouraged them to take it to the next step and run it in a dynamic model. I believe that if you regulate nutrients at a single critical condition, in all likelihood we're over-regulating, because that condition doesn't exist that long, and most of this stuff most chlorophyll and nutrients are responded more on an average loading condition, give or take impacts of meteorological conditions and retention time. So I think that it would be very conservative to use a steady state model to make a decision on benthic algae.
All right, a few more questions have come in. Does a two-dimensional hydrodynamic model assume instantaneous lateral mixing?
It depends upon which way the two dimensions go, right?
Right.
If they go vertical, if they go vertical, it's instantaneous laterally. If it's laterally, it goes instantaneous vertically. So, in most river systems, unless you're in a deep system, you're probably not going to want 2D vertical. You may want 2D, laterally, because you may have gradients across your river.
Okay, and can development of only nutrient endpoints be ecologically meaningful without developing DO and pH endpoints for the same water body?
I'm getting outside my lane a little bit here.
Yeah.
Yeah, yes, no, but yes, we would all recognize that you can develop nutrient endpoints that don't necessarily have to consider pH or even dissolved oxygen, and it's been done before, and actually EPA's eco-regions guidance that came out I don't know how many years ago, that was all done using data and a reference approach, so they didn't consider directly those numbers and how they affect dissolved oxygen or pH.
Right, yeah, sorry, that was a little outside of the kind of modeling focus of this webinar. So next question, is it
Great question.
Yeah, I think that's a complex one for the group listening, and I know EPA has done some other discussions about that, and I believe there's some other resources available for information like that. Is it possible to model nutrients, algae, or total organic carbon in a tidal portion of a freshwater river related to drinking water use attainment? And I think you're going to get to that later on, but.
Well, not specifically model carbon, but yeah, there are models that most of the bigger, more complex water quality models. Carbon's a state variable. You need if the model can do pH, carbon's pretty much closed except for the atmosphere, totally from the atmosphere. So yes, you can.
Okay, and then I think two more here, and we'll move on. The model calibration process is obviously an iterative process where you're tweaking multiple parameters and coefficients. And so the question is, which parameter do you generally calibrate first in a eutrophication DO model? Do you focus on something like ammonia, or then nitrate, or TP, or algae, and then go back, or is there an order that kind of maybe a general rule of thumb or something that you like to use?
I could go all day long on this. So let's start with the stream first again, because they are one of the better ones, easier ones to do this. So my approach, if I'm doing DO, the first thing I'm going to do is I'm going to do DO BOD, and I'm going to get those calibrated, by changing the one, given our laboratory abilities now, one, it would be kind of nice to know what the BOD decay rate is, because that can be measured in the laboratory, but we can adjust how we're doing reaeration in the system, and we should be able to get DO and BOD pretty much, in the ballpark. Now, if you know that you have large phytoplankton populations in your water column or benthic algae, you know that you're going to have impacts on DO from photosynthesis and respiration, as well as impacts of DO being consumed by when they die and converted to BOD. But the next thing I would do is I would go to the nitrification, and then if you have large phytoplankton or benthic algae in the system, then you go to the impacts of the phytoplankton on the DO. Not a simple process, but it's a very iterative one. One of the things people, when they try to calibrate these types of models, is they go in there and start turning all the knobs at once. It's best just to turn them one at a time, starting with BOD, then going to ammonia, and then going to phytoplankton.
All right, for the sake of time, we've got a few more questions, but I think we'll do one more and then move on. Can these type of models be used to simulate benthic algae in kind of mixing zones, or kind of, I guess, variable zones?
So that's a great question, because one of the things we didn't focus on is there's two types of, well, there's more than two types, but the two models we're talking about here is we have models that are capable of doing mixing zones, where it's basically a flume analysis. The one that's readily used here in the United States is called core mix. Another one is called plumes, visual plumes. And basically what that is most water quality models in a river are going to assume that everything is mixed completely across the river. So as soon as a discharge or a source comes into the water, it's mixed across the full width of the water body. Well, that may not be what's going on. So you can do mixing zone types analysis with these other water quality models, but it would require you to break your water quality up across the river, so that stuff is not mixed completely across. And probably the best way to do that is with a 2D hydrodynamic model and a water quality model.
Thanks, Tim. I think we'll wrap this part up. I'd just like to say there's lots of questions. Thank you, everyone. I think this really makes the webinar useful. Number of questions have come in that are kind of a little outside of the scope of this webinar. Several questions on kind of more policy, procedure, or endpoints for specific rivers. I'm going to kind of skip those, and for those of you who have asked those questions, there are EPA and state resources available. You could send me an email after this, and I could try and connect you with those people. But I'd like to keep the questions for today's webinar more focused on the technical modeling aspects and save the policy questions for more of an offline discussion where you can get an answer from some EPA folks who specialize in those issues. And with that, Tim, why don't you take it away?
Okay. All right, so we're going to move into modeling lakes and reservoirs. So as I told you, a river is the easiest one to model. Lakes and reservoirs are by far the most difficult ones to model. And a lot of it has to do with just their general characteristics. Basically, you know, lakes and reservoirs are standing water bodies, or they move slowly, or their flows are controlled by releases from dams. So they have a longer retention time. That gives more time for nutrients to exhibit impacts on the stressor things that we look at. So the velocities are much lower. What goes on out there is more, the mixing of the water column is basically driven by wind and heating that causes the water to either rise or sink. We talked about the residence time, so there's a lot of time for cycling of the nutrients, not only just within the water column, but also within from the sediments. So all of that becomes kind of critical. Lakes and reservoirs are typically much deeper than streams and rivers. Typically, they do stratify, and the main thing that causes stratification is heat. Heat causes surface waters to warm, and then a thermocline is developed, and at that thermocline where the warm water is above it and the cooler water is below it, there is less transfer of materials or vertical mixing during that period of time. Light does not reach the bottom of most reservoirs or lakes, so there's little activity in the lower waters. Reservoirs are much more common in the south than natural lakes. In the region that I'm in, I mean, almost everything we have is reservoirs. They have dams on there. They are controlled releases from the reservoir, whether it's peaking hydropower or to maintain navigation. So they're highly, highly managed. So the shapes of reservoirs are kind of critical in the types of models that you're going to select here, because you know, some of them, if they're run-of-the-river types of models, you can probably, a lot of them you can use you know, a 2D approach. If you have ones that have large, we call them wings or embayments off to the side, because when the dam was put in, it basically flooded a river valley. You're going to probably have to use a three-dimensional approach, because things are not going to be the same across the width of the reservoir. And you have to look at the important factors that control volume, you know, and residence time in the system. So where are your inflows coming from and such. So, you know, the typical, the stratification system in reservoirs in early spring, you know, they're completely mixed because there really is no thermal stratification at that time. In the summer, where you actually can get multiple stratification levels in the system, and that's where surface waters become detached from the bottom waters. In the fall, when the reservoir lake cools down, the surface water cools faster than the bottom waters, and they become more dense and they sink down to the bottom, causing a turnover in the reservoir, which can also have a profound effect on water quality. So all of this has got to be kind of taken into account when you model the systems. So for lakes, steady state models rarely use them. I mean, it would be very, very difficult to do anything with a steady state model. So dynamic analysis is usually required. Given where we are today with our water quality models, it's probably best to do whole lake, do the whole lake or reservoir, include it all in your modeling domain. For as far as how you break it out, pretty much almost all of the water quality models take a box model approach where it basically breaks your water quality up into segments, whether they're vertical or horizontal, and they're connected together. There are some one-dimensional vertical models that can be used. The Corps of Engineers uses a lot of them when they develop their water control manuals to look at impacts. You also can have two-dimensionally longitudinal and vertical models, but typically almost all of them that are done today are done with three-dimensional models or a specific model from the Corps of Engineers that has a long, long history of being applied to reservoir systems, which is a two-dimensional laterally averaged model, called W2. So I'm going to show you another example of a case study here where we're looking at impacts of nutrients, and the endpoint here is actually going to be a reservoir. And again, we're using LSPC to provide flows and loads, but in this case, we're introducing a three-dimensional hydrodynamic model called EFPC, where LSPC will be giving the flows to EFPC and the loadings to the WASP model. The EFPC model is going to be applied in 3 dimensions so that we can account for lateral and vertical variations within the reservoir. And then we're going to be using the WASP model to look at the impacts of the watershed loadings on water quality in the receiving water. This is the watershed that's being simulated in. You can see that it's mostly in the state of Georgia, but the receiving water body is actually in Florida. We actually set up 2 riverine WASP models that will provide that'll integrate the loadings and flows from the upper watershed to the lake itself. So there's 2 major arms. The first arm is called the Little River, and that ultimately comes into into Lake Talquin. There's another arm called the Ochlockonee, which comes into another arm. So the Ochlockonee River watershed comes in at this point here, from most of it's from Georgia. The Little River arm comes in here, and we'll be using the EFPC WASP model to predict water quality concentrations in this grid. So each one of these boxes represents a horizontal cell, and then for each one of those cells there's actually another layer underneath it. This is a fairly shallow lake. So in the end, we have a multi-model grid here that the green is our LSPC, our watershed model, and you'll also notice that the watershed model will provide direct loading to the lake from the surrounding watershed. The red network is the Ochlockonee River model, the magenta is the Little River model, and then the yellow is where we're interested in seeing where the nutrients are expressed. This model was actually run, the watershed model was actually run from 1996 to 2012. One of the things about using watershed models is you don't have any chance of getting them right if you don't get your meteorological conditions right. So we used a total of 7 rain gauges using hourly rainfall to derive our flows coming into the system. One of the things that's kind of critical is if you can't get the inflows right, There's no way that you can get the loading right. So this is actually a representation of the predicted flows and the measured flows at a USGS gauge from the LSPC model. And then ultimately, we're going to calibrate. So the watershed model and the two river models are run all separately, and they're ultimately linked into this system. So most of the study or effort was actually spent on calibrating the water quality model to Lake Talquin here. So the red dots represent where we have data for calibration. And again, using the same process I talked about before, the first things that we're going to do here is we're going to calibrate, we're going to calibrate solids, which is kind of important, PFS, because it affects light. But then we're also going to calibrate initially DO, then the nutrients, and then phytoplankton. So this is just an example of running a long-term simulation and to see the variability in the system. And you'll see when I get to a couple other graphs that things change dramatically from year to year. And you also, what this graph kind of shows you is that one in the upper left is actually the observed versus the predicted over the period of time that we had data for there. The upper right is just a probability distribution, which kind of shows you that the line is the model and the red dots are the samples, and that kind of shows you that the model gets the frequency and the concentration. The bottom right is just a one-to-one plot, and then you have some basic calibration statistics. But just to look at some of the dynamics in our system here, if you look, this is the chlorophyll response in the lake as a function of the loadings coming in. And I'll point out the graph up at the top is total nitrogen, and you can see when total nitrogen peaks into the system, one of the major components of sources of nitrogen is down here, which is right about here. When nitrogen comes into the system, you'll see a large response in chlorophyll. And then you'll see that, what's happening with phosphorus. And this is actually river miles, so that's 0 all the way up to kilometer 25 up here. So you can see changes. Now, being able to look at model results like this allows us to feel comfortable that, one, the model is representing the system fairly well, but it actually goes a long way to convince stakeholders in the system that you've got the major processes tied down. So one of the things that we were looking at is ultimately endpoints in the system. So for Florida has numeric nutrient criteria for lakes, and they're based upon the color of the lake and the alkalinity of the lake. So for our system here, it's a dark system. So it has a criterion. So one of the first things that was looked at is, well, how do you break up the lake into zones for assessment purposes? In other words, which zones behave appropriately or alike that we can make decisions for making our reductions. The other thing is that their numeric standards are actually based upon annual geometric means. So one of the things, and this may go to talk a little bit about a critical condition for nutrients, because it's very, very difficult to see. But the graph, the bar graph on the upper right, basically shows the predicted annual geometric mean for each one of those zones. And you can see that it changes dramatically from one year to the other, and it may be difficult just to pick a single, even critical year to do this. So you can see it goes from 2006 to 2012, and it changes dramatically. But it also does show you that there may be one controlling zone here that may need to be looked at. So we calculate the annual geometric mean for chlorophyll, TN, and TP. So if you meet their water quality, the American water quality standard for chlorophyll of 20 micrograms per liter annual geometric mean, you can set your TN and TP values to match that. Another interesting thing is, another way of looking at model results is, I don't break it up into zones, but use the model to help you to determined zone. Now, one of the issues about using models, of course, is that I can change the answer that we get from the model by changing my number of boxes. So we typically never use a single cell as a decision point in the model. Given the uncertainties in the models, we kind of want to use more than that. We want to feel pretty confident that it's not just a numerical anomaly for a specific cell. So what we're looking at here is we're looking at it by year, and any cell that is painted red basically says that 50% of that year that cell's predicted chlorophyll A concentration was greater than 20. And then you can see from year to year that the expression of the nutrients in the system changes. Whereas if you look at 2011, there's not much, 2011 and 2012, there's not much red, but that's because it was higher flow years, shorter retention time. For 2007, it was a low flow year, longer retention time. So we can use the model also to show us, well, the differences in from year to year, which takes into account changes in meteorological conditions and ultimately loading to the system. So that's one way that a water quality model can be used to help make a decision on what is the critical point in the water body to do a nutrient reduction for. With that, I'm up for questions.
Thanks, Tim. Great example for sure. I'll do another little pause here to give folks a chance to type in questions. We don't have many yet. Also, to do a little public service announcement, I think it was the Washington State guys emailed us to remind us that Qual2K apparently is now capable of non-steady state dynamic flow in conditions.
That is true, but it would be very difficult to do anything longer than a year with it.
So for folks out there, there's some links to Qual2K and the new descriptions at the Washington State modeling website.
Yeah,
and,
very good program to start using if you're new to water quality modeling, for sure.
Yeah, agreed. Yep. All right, we've got a few questions coming in. So I'll just start off then. Was Lake Talquin chlorophyll A output screen, I guess maybe I don't know what that means, an output from WASP?
So I'm assuming they're talking about the grids and the animations. That is live output from WASP. It uses a freeware utility that's part of the Water Resources Database program. If anybody's interested in looking at its capabilities, that's wrdb.com.
Okay, and maybe a question on resources. How do you know how much did it cost to model the Talquin example using the 3 linked models, or maybe just talk in generality if you don't have specifics?
Okay, yeah. So first of all, I got to give a shout out to my modeling team at EPA Region 4. That whole modeling process was done 100% at the region. The watershed model was developed by Dr. Glenn Fernandez, who's a hydrologist there. I would say it took him about a month or so to set it up and calibrate it. The Ochlockonee River Arm, we're a team player in Region 4. The Ochlockonee River Arm was developed by Amanda Howe. I was a little cruel to her because that was a very, very difficult system. That's 208 segments with 7 major dischargers in it. The Little River Arm was developed by Craig Hesterly at the region, and that has its biggest issue it has a discharger that has a flow control discharge limit. So getting that parameterized in a model is very, very difficult to do. I did the hydrodynamic and water quality models for Lake Talquin. Because that is the driver in this whole thing, what I thought I would be done with in a fairly short period of time. We've gone through 6 revisions, working with the stakeholders, and I would say that it probably has taken me about 6 months of my time over over 2 years. If that was to be contracted out to a contractor, you're probably looking at somewhere in the range of $400,000.
Great, thanks, Tim. Another question, how would you incorporate the effects of climate change into a model such as you presented here, specifically into a predictive model?
Sure. Basically, how you would do that is you would go back to the watershed model, and then you would change your meteorological conditions, which ultimately would change water temperatures, it would change runoff, so on and so forth. But the biggest uncertainty there is we don't quite know, how. Now, we certainly could do sensitivity analysis, changing temperatures and changing flows, or we could try to run a climate change scenario on that. And I've had discussions with a lot of different people. Given the time scale of the question that's being answered here is what are the appropriate permit limits for these facilities? Climate change is a decadal type of thing, and we're talking about a 5-year permit. So we didn't really consider it, but certainly the climate change scenarios could be fed into the watershed model.
Thanks, and I'll remind folks that in our 4th modeling webinar, which is available on the modeling website, it was about temperature, but it had a good bit at the end of it regarding climate change and the application of that in a model in Washington State.
Yeah, and that is a great example of how to do climate change and its impacts on temperature.
Another question, what guidance does EPA have for how to assess model output for decision points? And I think specifically you mentioned you don't use a single cell as a decision point, but how did you decide on the number of cells or which cells? Or
Yes, so I think when it comes to anything like this, the multiple lines of evidence is what you want. You're basically a car salesman at that point, that you have to basically say, hey, this is the way we kind of should look at it, keeping in mind that whatever decision you make is going to have to stand the public scrutiny. So we do not have a lot of guidance on that. I mean, how good is how do we determine how good the model is? Well, it typically is we run out of time or we run out of money. So we have to make a decision.
Agreed. Designated uses on Lake Talquin. Which designated uses were you specifically looking at?
So like on Lake Talquin, its designated use is fishable, swimmable, and they have numeric nutrient criteria for it. So that typically drives it. So I mean, that was, in the end, that's the when you have numeric nutrient criteria, it makes this process a little bit easier. Now, of course, how do you assess it and determine your reductions in the modeling scenario takes it a little bit further. But if you were to do that lake and you were working under a narrative criteria where you don't have numerics, the things that you would definitely be looking at is dissolved oxygen and phytoplankton abundance, and maybe pH.
Okay, another question. Does EPA have standard calibration metrics for evaluating water quality models and acceptable ranges of the same?
Yeah, when you run out of time or money, or somebody makes you give it. There was, I believe Tony Denisian, one of the authors of HSPF, published something some years ago where he had what he considered acceptable ranges for water quality in water quality models, watershed models, predictions to observe. I mean, there is nothing hard and fast. I mean, we're forced to make decisions with the best available information we have, and have to be able to defend what we do. But no, there's nobody. I can't tell you that if you get an R squared of this or an index of agreement of that, you're done, because it's never that way.
Right. With that, for the sake of time, why don't we continue on, Tim?
Okay. All right, so next, so I told you that lakes and reservoirs were the hardest ones to do, and the reason why they're the hardest to do is when we set up models, both hydrodynamic and water quality models, we kind of like to have gradients that we can compare the model predictions to observed data. Because if we're getting gradients correct, we can feel a little bit better about we're getting the right mixing processes in there. We have the right transport going on because we're able to replicate that. And in reservoirs, it's very, very difficult to, one, predict where the thermocline is, because again, in the two dimensions, the thermocline is moving up and down, but yet we have fixed cells, so it's kind of hard. Now, estuaries, you know, in the end, is where everything comes. So everything ends up in the estuary, and they have some impacts on it. Estuaries are pretty much dominated by mixing processes. You know, whether it is tides, changes in the amplitude of the tides, or it's wind. So, and tides change dramatically. This is tidal ranges of two different areas of the United States. The large tidal range is up in Seattle. The small tidal range is in Pensacola, Florida, and there's areas like Mobile Bay where its tidal range could be, you know, a centimeter or two. So these all control the processes. The other things that impact water quality is the bathymetry of the estuary. They affect the circulation patterns. It could be, for some of them that are fairly straightforward, things flow in and flow out and there's not a problem. In other ones where there is embayments and areas off to the side, you know, places like Chesapeake Bay, a lot of our bigger estuaries, you can actually get tidal trapping where materials get caught in these embayments because they get flushed up in there and they never get enough mixing or flow to flush all the way back out, and they get pushed back up in. So one of the big deals we have to worry about is tidal trapping, whether they move in or out. So that's a big, big thing we have to take into account with transport. Now, over the years, there's been many, many different approaches for simulating estuaries without using full-blown hydrodynamic models. And one of the principal guys that have developed a couple of different very good methods for doing that is a professor at the University of Virginia, Winston Lung, L-U-N-G. He's got books on how to do estuarine modeling using things like tidally average models, where you don't try to model every tidal cycle, but you actually model the effects of a whole tidal cycle over a long period of time. Then there are other ways where you can do intertidal modeling, which basically can do predictions within the tidal cycle. But what we typically do now is we use three-dimensional modeling where we're modeling the whole tidal cycle. So it's a little bit, well, it's probably the only way, or the only way you can make an informed decision now. So the big deal for the estuaries is you need to make sure that you are, just like the lakes, you need to make sure that you're getting your loadings correct into the estuary. We typically will bring our loadings in above tide, because once you're in the tidal regime, things are sloshing back and forth. So they'll come in above tide, and then we will, the hydrodynamic model is forced with a tidal range and a wind field, which takes into account both velocity and direction. I know there's a question screen here, but I'm going to continue on. So let me just show you some of the modeling approaches for an estuary and talk about the major components and the things that you have to consider. So we're going to use a hydrodynamic model, and you're going to use a water quality model. In my case here, again, it's going to be EFDC to WASP, but other models, CQual-ICM, which is the model developed by the Corps of Engineers, Carl Serco, that's applied to Chesapeake Bay. It uses CH3D and CQual-W2, so you got another 3D hydrodynamic model and a water quality model. linked together. And it's just so we can integrate all of that. So our potential endpoints for estuaries is a little bit more different. It's different than river to shore, somewhat different than lakes and reservoirs. So some of the stuff is the same. One of them could be a seagrass requirement, a light requirement to either restore or keep seagrass beds alive. As water bodies become eutrophic and chlorophyll concentrations get higher in the system, they cause impacts on light, and light hitting the bottom is reduced, and thus we get seagrass die-off. So a light requirement could be where you would control nutrients so that you control phytoplankton concentrations, as well as impacts of phytoplankton dying on DOC, which also scatters light. You could have dissolved oxygen endpoints to support sensitive species. So for a lot of you that are on the East Coast, you're familiar with the Chesapeake Bay modeling system, where they came up with site-specific DO criteria that support sensitive species, that make sure that there's enough dissolved oxygen in the water to not only have species survive, but also to propagate. You could have requirements for limiting anoxia or hypoxia. That could be controlling the total volume of water that is below a certain DO concentration, or it could be controlling the amount of dissolved oxygen in a critical habitat area. Another thing that you can look at is the community structure. In the estuary, we have a balanced flora and fauna. The food chain is not upset. I've worked on water bodies where high nutrient concentrations cause shifts in the phytoplankton species. They become undesirable species. The zooplankton that were feeding on them can no longer feed on them, so that you get a full change in the food chain. So these are some of the endpoints that you can consider for estuaries. So I'm just going to go into a case study in an estuary and point out some of the major things that when it comes to estuaries that you have to consider. This is going to be the Neuse River estuary. This is a TMDL I worked on. Well, it's about 15 years ago, but it still kind of uses the same approach that I typically try to use, where I use long-term data and it's a long-term simulation. For this thing, for the loadings up in the watershed, there's 32-point source dischargers. We have a lot of data collected. There's non-point sources up there. There's a lot of agricultural components up there, both row crops and animal feeding operations. We have impacts on atmospheric deposition. So ultimately, these are where this is the watershed, all of the green. All of this was not modeled in the TMDL model. The TMDL model was modeled down here at the bottom, just in the estuary itself. We just had the loadings from here. Ultimately, the goal of the TMDL was to reduce nitrogen coming into the Neuse River estuary to meet a chlorophyll A target, or actually a state standard. So much like Lake Talquin, the Neuse River was broken up into segments. There was, I think it's 420 horizontal cells here, and there's 4 layers in the model. Everywhere that you see a red dot, there was water quality data collected. One of the few modeling studies in my career where, one, somebody asked the modelers what we needed so that we can model and calibrate our model to, and 2, they went out and got it before we started the modeling. So this just shows you the modeling, the monitoring network. So the major things that were monitored is water surface elevation is very, very critical. That's the driving force of transport in the system. So we need water surface elevation somewhere in our model network, preferably down at our ocean boundary side. We needed salinity. Salinity is one of the critical things in estuary models that you can use because it typically has a gradient. Not only does it have a vertical gradient, but it has a horizontal gradient. And if you can get that gradient correct, you can feel pretty certain that you got the transport correct. So then we also need flows, and then we need the nitrogen and the phosphorus series. We need chlorophyll concentrations, CO concentration, sediment oxygen demand, and nutrient fluxes. The big environmental conditions that you need is you need something to predict light, or light coming into the system, temperature, and then the bathymetry is critical. As I talked about earlier, the morphometry of the estuary is critical for getting the transport correct in the system. So just to give you an overview, this is the Neuse River estuary. You can see that it's a fairly shallow system for an estuary. It really doesn't have a maintained navigation channel. One of the things I do have to point out to you is the Neuse River estuary is actually the widest river estuary in the United States. Down here at the bottom, it is over 11 miles across, so it's a fairly big system. So I just want to show you one of the phenomena that we have to think about in estuaries, and also the decision of how many dimensions do I need to consider. You know, can I do it one-dimensionally, two-dimensionally, or three-dimensionally? And one of the first things that I just want to point out here is just looking at some monitoring data for salinity, we can see something that looks a lot like this. So the green line is salinity at the broken green line, which is the lower one, is salinity at the top. The solid green line is salinity at the bottom. And then what we see here is there's periods of time like this here where the system is completely mixed. So the salinity in the top and the bottom is the same. So this is a kind of a critical thing that the model needs to be able to get, because conversely, there's periods of time where there is significant stratification between the top and the bottom. So when do you think it is that we have the greatest influences of nutrients on chlorophyll in the system? It isn't when the system is completely mixed. It's when it is stratified. So it's critical for the model to be able to predict or account for that stratification. So another thing that we need to look at is, so that's just looking at top to bottom at certain areas. Now the next thing we're going to do is we're going to look at what is it across the estuary, what happens across the system, and what are the critical things that control water quality there. So what we're looking at is we're looking at this is two stations, LT9 and S4. So if you go back here, it's these two stations. Now they look like they're relatively close, but they're a couple of miles apart. What we see here is this is salinity on the bottom. So we can see for S4, the salinity is going down, but on the other side of the estuary, salinity is going up. And this was a major phenomenon that had to be accounted for in the Neuse River, because one of the things that they experience there is fish kills. What happens every day in the afternoon is the wind comes up on this system and it blows the surface water across the estuary. Well, once the water is pushed to one side, it's got to displace water on the other side, and what happens is the lower water on the other side, where you see the salinity going up, is upwelling, and that's causing hypoxic or anoxic water to come up, which traps the fish. So again, this is a kind of a critical thing for setting up this water quality model, and pretty much gives us good confidence that we should probably be applying a three-dimensional model to it. So the approach is the approach here is to set up the EFPC model and calibrate it for flow, water surface elevation, water temperature, and salinity, because that's how we feel confident that the transport's done correctly. Once that's done, it's passed on to the water quality model. So just to show you how dynamic the Neuse River system is, and this is pretty typical of just about any estuary, the top panel is actually looking down the middle of the estuary, so that's a side view of the estuary, and we're looking at salinity concentrations. To the left is the freshwater inflow. To the right is the ocean boundary or the Pamlico Sound boundary. The middle graph is showing you the bottom cells looking down on the Neuse River estuary, and you can see where the salinity is changing. You can also see where the area of where fresh water meets salt water. And then the bottom two ones are just showing you a cross-section at those two stations to see how salinity is changing laterally in the system. Most of the time in estuary systems, chlorophyll's biggest influences or highest concentrations are going to be in areas right in here, because this is basically the where the retention time is the highest for the freshwater component, but yet the salinity concentrations aren't high enough to kill the phytoplankton. So it's a very dynamic system. In the case of the Neuse River, the transport system is actually mostly controlled by wind. It's actually the sailing capital of the mid-Atlantic states, so wind is a critical component. So once the EFDC model is set up and calibrated, the model is fed forward into the water quality model. And I will tell you that the success of the water quality model is 80% because we've got the transport totally correct. Never mind the loads, but the transport. And all I'm going to do now is I'm just going to show you some of the results from the water quality model and just talk about some of the things that we're looking at when it comes to an estuarine system, how we feel that we get pretty good confidence the model is doing well. So we're going to start it towards the top of the estuary, and we're going to look at monitoring stations. And again, there has been no other water quality modeling study that I've worked on in 25 years where I had this much data to help parameterize and calibrate a model for. So we're starting up at the top. The first one is salinity. Now, salinity and temperature are predicted by EFDC because they are critical components in the calculation of the transport. But one of the things that we feel very, very good about is because this is up towards the freshwater section of the estuary. We can see for most of the time there is no salinity, but there's small periods of time that we get salinity up there, and the model does a very, very good job of showing that. What we take from that is that, and this is a 2-year simulation. What we take from that is we take that we got the transport correctly. Because if our boundaries or our forcings weren't correct, we would either not have enough push from the ocean side to get these spikes in salinity. Temperature is by far one of the easiest things to model in an open system. I will not argue with my colleagues in the Northwest where they have to deal with shading, bank shading, and all that stuff, and they're trying to make calculations on 1/10 of a degree. But it does temperature very, very well. So there's two gradients that I looked at on the Neuse that made me feel very, very good that the model was doing well. The first one was the salinity that shows temperature, but the next one that I felt really, really good about was the nitrate, and there is a clear signal of nitrate gradient in the system, and the model does very, very well because the major component of nitrogen in this system actually is nitrate, and you can see that it gets the seasonal variations, it gets the lower concentrations, and then the recovery going back and forth. The other gradient that we'll see here is not only are we getting the temporal gradient, but as we move down the estuary, you'll see that the nitrate gets attenuated and it's going down, and the model does a very, very good job at simulating it. Phytoplankton, it does a pretty good job of doing as well. This model, there was only one phytoplankton group considered, so this is a homogeneous representation of all the different species out there. So as we just move down a little bit further, you will see that salinity is going up. We're still getting that nitrate signal. Everything is really, really good. One of the things I want to point out in this thing is because it's also one of the utilities of a model is if you look at this period of time here, you'll see that we had almost 15 parts per thousand salinity at the station, and it went down to 0. Well, that was caused by Hurricane Floyd, which came in and it dumped a lot of water on the Neuse River Basin, went back out to sea, and then it wasn't done, and it decided to come back and do it again. So it actually shows you that the model was very, very sensitive to those changes in freshwater flow and a large perturbation to the system. And likewise, the water quality model's response to the changes in loadings of those times was pretty good. So, and this is just taking us down a little bit further. You can see the nitrate's going down, but we're still getting that seasonal signal very well until we get down to the bottom. So that's so for the estuary models, it's critical to get the transport right, and the only way that you can get transport right is if you have a gradient to compare it to. There are other techniques that are more readily available now also to make sure that you're getting the transport correct in an estuary that weren't available back in 2000, or at least readily available. Is we have the ability to either mount or drag ADCPs that can measure velocities over depths. We can use that to compare to what the model is predicting to make sure that we're getting the transport as well. So the critical take-home for estuaries is you really do need to nail transport. If you're not getting the transport right and you're calibrating the model, you're probably trying to make the model do something for all the wrong reasons. And if you're interested in more information on the Neuse River modeling effort, this is a great publication because there was 3 other modeling approaches that were going on the Neuse River at the same time. They were all published in this same journal where they are compared and contrasted. With that, I just have one more thing before we take questions, and I just wanted to, this does not represent all of the models, and I did forget to check. I should have broke qual 2KW out. It does do time variable, though it's only really capable of doing about a year at a time, but this shows you the different models that are typically used to look at nutrient impacts, from the bathtub model to the 2 core models, C-qual ICM, W2. EFDC is both a hydrodynamic and a water quality model. EPD RIV1 is a modernized version of the core's C-qual RIV1, which is a very good one-dimensional hydrodynamic and water quality model. And then you have qual 2K and qual 2KW, and then WASP or WASP and EFDC together. And the big things I wanted to just point out to you is the ones that are time variable, the ones that are steady state, how many dimensions the model is capable of handling, can it predict yield variation in the system, and then the major components that you want to look at, which is dissolved oxygen, algae, the ones that have the ability to simulate more than one algae, the one that can do the benthic periphyton algae, the ones that can do macroalgae. One of the critical things that we talked about in the previous webinar is the ability to predict light, because as we go through management practices and nutrient reductions, we're changing the light in the system. And for almost everything, at least we do in the southeast, light is actually the most limiting nutrient to phytoplankton. So we actually put best management practices in that reduce nutrients, but also increase light attenuation, and we can actually make it worse. The other thing to consider is the impacts on pH and alkalinity and the models that can do that. And then the other big one that's become relatively important, and because the capability exists, is the ability to predict sediment diagenesis, which is the decomposition of materials in the sediments that cause an oxygen demand and also a nutrient flux to the overlying water column. Again, as we go through reduction scenarios, we need to account for changes in SOD, oxygen demand, and the nutrient fluxes. So these are predictive tools based upon the fluxes of nutrients and carbon to the benthic segments. And then for some of the nutrients or for light, it may be important to do sediment transport, where settling and resuspension is a function of a mechanistic approach based upon particle sizes and velocities. So these are the typical tools that are used now for continuous simulation and steady state simulation for nutrients. With that, I'm done.
Great, thanks, Tim. So I want to start off by first giving you a huge thank you, Tim. It was a great presentation, and giving the Region 4 modelers a big shout out. For those on the call today, the Region 4 team does some phenomenal work within EPA. They have a number of publications, TMDLs, presentations online that you could search for. Done some great work throughout the southeast U.S. and I encourage you, if these are things that you're interested in learning more about, to simply look up some of their work. It's top notch. All right. So to launch into the questions, then. First question, I think you just touched on this at the end, Tim, but this came up earlier, is could you discuss, modeling sediment deposition and then the release of nutrients? Maybe get into a little more about how detailed of a job you get into when looking at the various scenarios that you presented.
So yeah, that's one of the biggest rubs, right? Is depending upon your systems in streams. Free-flowing streams, it's a lesser of an issue than it is in lakes and reservoirs and estuaries, where you have a longer retention time and you have larger areas of net deposition. So, prior to the development of the sediment diagenesis model by Dominic Totoro, what we've kind of relied on was field measurements. And actually our Region 4 Field Services group actually developed a protocol for measuring SOD and nutrient fluxes in situ by using chambers. So we were kind of relied on, in the past, on, well, what were these chambers, what was measured, and we would kind of apply it. Now, one of the difficulties in applying measured data is it's not measured everywhere, but you have to make an assumption and apply it everywhere in your models. So it was a difficult thing to do. And then the next thing is, if you did a nutrient reduction, you'd have to determine, okay, well, what is the change in the sediment dynamics? And it was very, very difficult to do. So with the development of the sediment diagenesis model, it has allowed us to basically calibrate the sediment diagenesis model to the available measured data that we have, use it to predict spatially and temporally the sediment oxygen demand and nutrient fluxes. Keep in mind that the sediment oxygen demand is a flux of oxygen demand from the overlying water column into the sediments, but the nutrient flux is a two-way street. Sometimes it's down and sometimes it's up. And depending upon the loading history, the age of a reservoir, you still may be in a situation like the case of Lake Talquin, because Lake Talquin wasn't that old. The nutrient flux in Lake Talquin is actually a net down, and we can see that both with the diagenesis model and looking at if you did a nutrient balance of nutrients in and nutrients out. But for other systems, it's it changes as a function of how much is to be depositing and how much of it's new stuff and versus old. So the sediment diagenesis model is a critical tool for us now to link changes in sediment demands to load reductions.
Great, thanks, Tim. Maybe I'll pause for a second again. We don't have a ton of questions, so this is the end of the presentation. If you have some questions, please get them typed in as we kind of go through the last logistics here, and we'll finish up with that. But first of all, I just advanced the slide to show the participation certificate. There is a certificate available for today. You'll get an email also at the end of this presentation, or maybe tomorrow perhaps, that also has these links on this page, so you don't have to go scribbling these long URLs down right now. Future webinars and again, the past archives are available at the link at the bottom of your website there. And with that, we'll get back on to some additional questions. Tim, the Neuse River model predicted, the Neuse River predicted DO appeared to over-predict the minimum levels, and the asker is referring to slide 63. How does potential bias and minimum DO predictions influence model interpretation?
Yeah, okay, so DO actually was not a major concern for us, and we actually really didn't even calibrate to it because it was not one of the endpoints that they were interested in. Certainly, certainly we could have did a little bit better on that. That's for sure. But yes, I totally agree with that, and we knew it all along. But again, it wasn't one of the endpoints. It was actually one of the endpoints the state didn't even want us to look at.
And Tim, we had a question. You posed that last slide that had some examples of the different models that would apply to an estuary/lake system. And we got a question on, could you discuss some of the models that apply to watershed nutrient modeling? Just maybe tick some off the top of your head for folks that.
Well, I see the question, and I'm reading that a little bit differently. I'm not quite sure if
Okay.
Paul wants to weigh in there. Is he asking, can I discuss watershed nutrient models? In other words, comparing the water quality components of like HSPF to that of a water quality model. I think that's what he's asking.
Okay, sure.
But I'm not sure. Yeah. So you know, HSPF, LSPC, SWAT. They all do have the ability to predict nutrient cycling in the aquatic environment. Their capabilities are not as robust as they are for the water quality models that you know, have been developed through the years. So yes, they can be used. It really depends upon what your ultimate goal is. If my ultimate goal for Lake Talkman was just to get the total nutrient load down to the lake from the watershed, I would not have needed to use WASP up in the river networks. The reason why I used WASP up in the river networks is we went up with the detailed water quality model to collect the point source discharges. And to be fair to them, their nutrients would cycle exactly the same way as they would in the lake, which is more of again, it's a more robust water quality model. So yes, you can use the water quality components of the watershed models, but they are kind of, you know, well, they're more simplistic.
And Tim, maybe I'll ask a question. For the last example that you just presented, you showed, you know, the variety of models that you could choose from for the Neuse River estuary, and, you know, their capabilities. Can you get into why, or, you know, why you picked EFDC versus one of the other ones?
Okay, well, actually, that table was not for just the Neuse River estuary. That was water quality models to do any type of water body. So on the Neuse River, there was, and if you go to that journal article, it explains to you, there was 3 different approaches being taken there. One was a Bayesian multi-regression model where they were going to, you know, set up this Bayesian model, regress against the available data, and then determine what kind of reductions would be needed to meet the water quality standard. I can go back and I'll talk about the weakness of that in a second. The next one was before EPA got involved, there was a CQual W2 model that was applied to the Neuse River. And one of the things about CQual W2, it is a very, very good model. It's got great capability. And one of the issues is it's 2D laterally averaged, so that meant water quality was going to be averaged across the estuary. And I've already mentioned that the estuary is about 11 miles wide down there. And that would mean that nutrient concentrations on one shore would be the same as nutrient concentrations on the other shore. And we had seen in the data that that's not the case. There is a gradient going across the estuary, so it's kind of critical to do that. So that left us with choosing a 3-dimensional approach. When it comes to the 3-dimensional approach, there's really only a couple of choices out there. You know, when I talk about models too, I also talk about models that are available in the public domain and readily available to anybody. You could have used EFDC and EFDC's internal water quality model. It's a capable model and it can be done. You could use CQual ICM linked with CH3D or EFDC, because it can do either. Or in my case, we chose EFDC and WASP. So, but that table was for all, you know, that's basically the same capabilities for all. Now, so, I believe that the 3D approach was the best approach there. The 2D model had issues. Some of them we were actually able to fix by giving them some of the results from the 3D model. This is the issue with using regression models, especially when it comes to nutrients, is if you come up with a regression model based upon the data that you have, and ultimately your reduction is going to take you outside of the data that you use to develop your regression, you are in some very uncertain regime there. That's why I think it's a little bit better to use a mechanistic model where we have relationships within, physical, chemical, biological relationships that can actually relate changes in nutrient concentrations to chlorophyll.
Great, thanks, Tim. I think Paul chimed in with some clarification on his question. Said you mostly showed in-stream models, some examples and their capabilities of watershed models, and the ones he lists off are WARMF, WARMF, HSPF, etc.
Right. So, I could talk a little bit about WARMF. I've got a little bit of experience with it. WARMF is a model that was developed by the Electrical Power Research Institute. Its origination was actually to support the acid deposition program. It's an overly ambitious tool where it probably tries to do more than it can, but it does have some very good capabilities for hydrology and nutrient cycling, but it also tries to be the end-all, be-all for everything, which is very, very difficult to do. One of the things that you can see is I kind of believe in the linked approach, where I take the watershed model, the hydrodynamic model, and the water quality model, because none of us modelers do it all, and this allows me to pick the best of each and use it in making our decisions.
Yeah, thanks, Tim. With that, I'm going to advance the slide. There we go. Just another public service message while folks are getting their last questions typed in here. Our next webinar, and you may remember we originally had just planned 6, but we did get some additional funding, and thank you to EPA headquarters and ORD for that. And so webinar 7 is going to be regarding sediment modeling, and I think we're going to set up the next 2 webinars, number 7 and 8, very similar to what we've done for the nutrient webinar, and that webinar number 7 will be about kind of more sediment basics, and then webinar 8 we'll get into some of the more details and examples of sediment modeling. Don't have any specific dates on those yet, but I do know we're going to take a couple months break here through the holidays since folks are traveling and not generally available around Thanksgiving and Christmas. But an email will go out to anyone registered for any of the webinars advertising the next one as soon as we know the dates, and you can always check back on our modeling website that we have here on the screen. And with that, we definitely have some time for more questions if you're up for it, Tim.
Oh, sure, why not?
Question about nutrient load released from the sediments. How do you estimate how much of the nutrient load released from the sediments reaches the photic zone where it can be used by primary producers?
That's a great question. Boy, that would take a lot of post-processing and digging into the model. Oh boy, so one of the things I guess you'd have to do it by difference. The models do output the total release, grams per meter squared per day. So you can you'd have to set up and do you'd have to dump the information out of the models and set up a mass balance to see where it is coming from. One of the things that we're hoping to add to WASP in the future is a flux output that'll output all of the different fluxes, so you'll know where the nitrogen came from, where it went to. But the big deal is how much of it's coming up into the photic zone. That's a different thing, because that would almost require separate state variables for all the sediment stuff that would get added to later on. So we're not quite there yet.
Thanks,
That's
Tim.
a challenge, though.
Kind of a WASP-specific question real quick. What solid type did you use for the nutrient setting in WASP segment solid 1, 2, or 3? I don't know if that makes sense to you. It doesn't mean a lot to me.
Well, let me say hello to Luke now. I've been working with him up on Narragansett Bay. So, I think, Luke, I need to just talk to you directly, because the nutrients themselves settle without using the solid types. So the solids are you know, inorganics. So I'll talk with you. Thanks.
I'm going to advance one more slide for another public service announcement. This is just the 5th reminder, I think, about our water quality modeling website, where everything is posted and archived. We hope to be updating this soon, so more information to come on that. Also, at the end of this presentation, you'll be given an opportunity to provide feedback on the webinar today. We encourage everyone to do this. We do read every bit of feedback that we get, and the feedback that we do get is used to figure out what we're going to present on for future webinars. So really take that seriously. So please take a minute to fill that out. And not many more questions coming in, Tim, so I think we've got just one more, and then we'll shut things down and let people do the survey at the end and end a little early. So the last question is, could you give your opinion on water quality modeling using GIS software packages? And the asker doesn't give any examples. I'm not even sure if there's anything out there that would work, anything that you know of.
Well, I mean, there's a host of, not so much water quality, but water or flow-based stuff that does GIS. Now, I mean, GIS software is absolutely critical in everything that we do. Embedding the software inside the GIS, somebody just popped up ArcHydro, which is a prime example of that. One of the issues that you have running tools like that inside the GIS is it's not the most optimal platform for running these models in a timely fashion. You know, a model like the Neuse River model, it takes about 4 hours to run a year. The Lake Kaufman model takes about 40 minutes to run 6 years, but if that was inside a GIS system, it would take probably 10, 20 times longer. I think the biggest aid that we get from GIS is for visualization and processing geospatial information and giving it to the model. So we have a model builder inside ArcGIS that builds all of our LSPC inputs based upon subbasin delineation, land uses, soil types, so on and so forth. So we use it indirectly, and then we may use it indirectly on the back end to help convey results. I am a big proponent of EPA's Basins tool. The Basins tool actually has in it tools for building several different models. SWAT, HSPF, it has a tool in there to build Aquatox and WASP model networks. So it's a great tool, and it's one that is very valuable and helps us do our work in a more expeditious fashion.
Well, I think with that, we're going to end the webinar. Just wanted to give one last big thanks to Tim. Great job today. Thank you to everyone for participating. I hope to see you back here for future webinars. We'll keep you posted on those. And with that, I think we're going to go ahead and close out.
Thank you, Jason. We've got a great thing going here.
Yep. All right. Bye, everyone.